Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. Thursdays at 7, we'll see you there.
Do you say that you can even do live imaging with...
Yes.
Okay, so what does that mean, live imaging for humans? I would give you my arm. Could you actually live image my skin?
Skin is the best part of the human organ. It's the largest organ, and it's actually a very important organ tissue for steady aging processes. The light doesn't penetrate much into the deeper level of the organ. For example, if you want to see a liver, it's very hard because that penetration is limited for a few hundred micron. So the skin is perfect.
How deep can you get in?
So it's a few hundred micron. It's like a 2-photon fluorescence. The golden window can make a little bit deeper. From 1600 nanometer to 1870 nanometer window. That window can reduce the scattering in the tissue, and you can penetrate even deeper.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us today. You just saw a brief summary of our last episode with Associate Professor Lingyong Shi talking about live imaging to help us understand metabolic dynamics and aging. And I can tell you, we not only talked about the skin, but lots of other things. So watch the YouTube episode on our YouTube channel. Today, Assistant Professor Peter Lidsky is with us. And have you ever heard about that evolution could kill? Actually, evolution might kill. This is going to be discussed today, and I am for sure this is going to trigger many questions. So please use the Q&A function to send your thoughts. Dr. Ayla Hotzkut-Kuric, our Senior Research Fellow, will moderate your questions, and she will introduce herself first.
Hey there, could you tell me a little bit about yourself?
Hi, I'm Ayla Hotzkut-Kuric. I'm a physician scientist specialized in medical pharmacology. Here at NUS Academy for Healthy Longevity, I design and, together with the colleagues, lead clinical trial that tests the effect of multiple drugs and dietary supplements on the biomarkers of aging, all with one aim: to make people live healthier for longer.
That's amazing. And what are clinical trials, and which one do you lead?
So the clinical trials are structured studies where participants volunteer and conduct various assessments. We collect the data, analyze it, and like this, we can assess the effect of various interventions.
Thank you so much for explaining. So what is it like being a participant? What do they do when they first arrive?
So my colleague Vanessa already showed our new clinical trial center. However, now it's ready and full capacity working. So come and let me show you. Hi, Ding Ding.
Hello, welcome.
This is Ding Ding. She's our clinical trial manager, and she makes things work smooth from registration to the completion of the trial. So first, our participants get registered.
You're welcome.
Hello, good
Can I
morning.
get your ID card with me? Okay, thank you. This is for registration.
So after the registration, our participants are going to be briefed about the study and assessments that are going to be conducted. We're also going to ask them to sign the informed consent. Come, let me show you.
So I'm going to tell you more about the studies, so let me know if you have any questions.
Thank you.
So after the informed consent is signed, we proceed with the collection of biological samples. Let me show you.
Okay, so now we will do a blood draw for our participants. Okay, so you can put your hand over here. You'll just feel a slight pinch and then it's done. That's it. Yeah.
So this is where we do our anthropometric measurements, such as measurements of body weight, body height, the BIA assessment. We also conduct other tests, such as measurement of hand grip strength.
This one is the hand grip machine, and we will hold it like this. It should be 90 degrees, and those hands we will test 3 times. Okay, the first time for your left hand. Okay, 3, 2, 1, go. Squeeze, 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 squeeze. Carry on, carry on. Yes, relax. Okay, very good.
We also conduct a cardiopulmonary exercise test to measure your maximum oxygen consumption. Let me show you. We also assess the effect of our investigational products on skin. Let me show you. We have many other assessments, like cardiovascular, ophthalmological, but we are going to leave this for your longer visit. Perhaps you can be a research participant in our trials.
So thank you so much for that tour. I really enjoy it. Could you tell me which clinical trials you're currently running?
The first trial I want to talk about is the CERT6 activator study, where we assess the effect of fucoidan, one compound found in brown seaweed. We want to see what is its effect on biological age in middle-aged to older men. So this is one of the studies. Another study is a Prometheus study. With this study, we are actually competing in the X-Prize Health Span competition. It is a multimodal study where we assess the effect of lifestyle interventions, dietary supplements, and physical exercise in healthy middle-aged individuals. And another study is the Accurate study, where we assess the effect of NMN, and we actually try to personalize the dose of NMN based on NAD values. We're using one AI tool called Curate AI. Also, we are assessing the effect of this compound in middle-aged to older individuals.
Wow, that's so cool. Ayla, tell us, what do you like to do outside of work?
I really enjoy the nature, and I love to do yoga.
That's amazing. Thank you so much, Ayla. How can we learn more about you?
You can follow me and connect with me via LinkedIn. I'm pretty active there.
Awesome. We'll see you soon later at the moderation.
Thank you.
Thank you, Ayla. Beautiful. So I think if you're living in Singapore, just check the website longevityacademy.sg and come to Ayla's trials. So, but today we are talking about evolution and if evolution kills. I'm honored to now introduce Assistant Professor Peter Lidsky from the City University of Hong Kong. And he is a virologist whose research explores virus-host interactions, genetics, and the biology of aging, of course. He completed his PhD on picornaviruses, so very small viruses, the biology at Moscow State University, and continued his research at the University of California in San Francisco, where he developed the pathogen control hypothesis of aging. I'm sure we will hear everything about it, a concept that now underpins his laboratory work in the evolutionary mechanisms of longevity. So, Peter, is evolution killing us?
I believe so, yes.
The floor is yours. Thank you for being here.
Thank you very much for this invitation. Big honor. So sure, I think I need to share my screen that shows my my title. It was a little bit gentrified by the team, and you probably are wondering what is besides me. So this is a cover of my book that I wrote recently. So it should be out like in some weeks. So, and if I will not manage to convince you that evolution kills in my talk, you may refer to this book. So, but let's start with the question. What is the main bottleneck in aging research? Because we are researching it for decades and we still don't have a good remedy. And I think the main bottleneck is that we don't know what aging is. This is a work of Vadim Gladyshev, who just surveyed different experts in the field, asking them very basic questions on basic definitions. For example, how do you define aging? And they say, no, well, something goes down, declines, so there is no consensus on even this very basic question: what causes aging? Well, some damage, something got damaged. Well, how do you define rejuvenation? So no, something goes back to good thing. What is the most important unanswered question? It's aging itself, right? So this seems to be we don't have a paradigm, we don't have a language in which we can speak about aging. And if we are cruel to ourselves, until this paradigm is developed, we may say that the gerontology doesn't exist as a coherent field, because it's more like a number of fields. And there are some labs that are trying to develop the paradigm, develop this language, and we also are working on this, our own Esperanto. And some people approach it from mechanistic side. We belong to evolutionary group. Why do we believe in evolution? No, because there is this famous quote from Theodorus Dobzhansky that nothing in biology makes sense except in the light of evolution. Our bodies were shaped by evolution to spread our genes, and this is the most powerful tool we biologists have to understand things. Mechanistic systems, on the other hand, are super complex and leave so much degrees of freedom that it is possible to build a non-contradictive model of aging around almost every process, almost every gene, and it takes ages to disprove these models. Oxidative theory of aging from Harman took 3 decades to get disproved. So, and if we look back into the previous success that led to dramatic improvements in healthcare, it always was that between understanding of the phenomenon and finding its physiological mechanism, there was evolutionary or ecological model. So, for example, infection was rationalized as a colonization of an organism with a pathogen. And with this, we came to antibiotics and vaccines, because we understood what to search for. Diabetes is we need to regulate sugar level while we eat sparsely, and this system gets broken, and that led to discovery of insulin. And for aging, unfortunately, this adapter, evolutionary or ecologically, is missing, we believe. And that's why we cannot find and tackle the mechanism. And but additionally, if we add one more PC in this graph, I would like to say that we are very different from all other labs listed, because we are the only who believe aging is a program and not the result of damage and entropy. What is this dichotomy about? So there are 2 main ways of thinking about aging. One is that aging is accumulation of damage like rust or friction. And another one is that it is a genetic program that kills us. Most scientists believe in this, as I said. Why they do so? So the first, before I explain why they do so, I want to say that these 2 opportunities are completely exclusive, and they define largely the way how we cure aging. So because in this case, we need to help these dots to go back in their places, so we need to fix something. In this case, we need to find this gun and remove the bullets. And this is it means that we need to find the genetic program and break it. So these are 2 completely different projects. If we invest in the wrong one, we lose all the time and money. And the reason why people believe in entropy is not because we have proved it experimentally. It is still not demonstrated and not supported by data. It is because we have 2 very strong evolutionary philosophical arguments of why program cannot exist. So, and these arguments are that it's not clear what the idea to kill the animals. And second, why don't we see immortal mutants? We will discuss these questions in the later slides. I would like to say that there are these canonical evolutionary models, antagonistic pleiotropy, disposable soma. To me, they are very flimsy. They support the entropic view, more or less. But I don't have time for now to discuss their weaknesses. And I go to the first question that we answered with our model. So I built a model that can answer to both these questions positively. So that let's ask ourselves, can death be good? Of course, it seems no, never, because our idea of evolution is replication. If I am dead, I cannot make babies anymore. However, there is a very well accepted situation when death is definitely good. So this is called abortive infection or ABI system in bacteria. When a bacteria is infected by a phage, there is no chance it will recover. So it wants to kill itself and save the brothers, which are its relatives or even clones, from these infections. So it sacrifices self to prevent phage from reproduction. And this is very well described, part of bacterial immunity. So the question is, can something like this also exist in higher animals? And we believe that yes. So our model is that there are sterilizing chronic infections. Sterilizing in this case means that the infected animal cannot produce babies anymore. So it can produce only these infectious particles. And also this infection should not be removed by immune system and should not kill too much. And then idea is that this animal is already useless for evolution because it cannot do babies, right? In human populations, examples of such diseases can be these sexually transmitted sterilizing infections. So this guy is committed to live to die without any progeny, but before doing this, it can infect other members of the population. And these other members of the population can be its relatives. And with this, this longevity of this infected individual can be detrimental for its own genes. Scientifically speaking, its inclusive fitness is negative. So this is an example from real nature. So this is a fish, this is a parasite. As a fish grows older and older, you see that more and more fishes are getting infected. By age of 8 years, almost every fish is infected. And so if this pathogen is sterilizing, then evolution may want to kill every fish after this age. So, and we can model this mathematically. So this is also a philosophical evolutionary work. So we built this model of viscous populations, like territorial animals that, like these wolves in Minnesota, like they move on the surface, rejecting each other. And this is a model that allows to recapitulate these kinship gradients. This is an aspect of animal populations, which is quite general, but is typically overlooked by classic epidemiological modeling. So it means that related animals are interacting with each other more frequent than just random members of the population. And this factor, we believe, is key for evolution of lifespan. Because I don't want to save, with my adaptive death, I don't want to save everybody on this Zoom. I want to save my children and my genes. This is idea of kin selection. So, and this is how the model basically works. So if we have short-lived animals, these yellow dots, and we introduce a long-lived mutant, then long-lived mutants are living longer, replicating more, and taking all the population. If we introduce pathogens, these black dots, in the pure populations of short-lived or long-lived animals, you see that long-lived animals are much more infected much better as a population because they spend much more time in an infected state, while short-lived are dying shortly, and with this the number of infected individuals is reduced. Now, if we assume that this infection sterilizes the host, so they produce 10 times less babies, then what will happen? So we again introduce this long-lived in a short-lived population. You see first it expands, but then the pathogen comes into this population of long-lived individuals, then it can remove it completely. And this short life can win over the long life. And with this, we answer the first question that was a problem for evolutionary biologists for a long time. So we believe the shorter lifespan helps to control epidemics of chronic sterilizing diseases. And this can make aging adaptive. But now the second question remains. Why don't we see mortal mutants? If aging is a genetic program, then it should be encoded in our genes by definition. So then by killing it, by mutating, inactivating these genes, we need to produce non-aging mutants, right? So in principle, we see some phenotypes that we can interpret as gain-of-function mutants. This progeria, this is accelerated aging. But we never see the immortal individuals Voldemort. And this is a very strong argument that we also managed to resolve. So, no, first of all, we need to state that such mutants are possible because we have mammals with negligible senescence, naked mole rats, and naked mole rats have common ancestor with squirrels or rats. And so it means that along this branch somewhere, an aging mother produced a pup with negligible senescence. So these mutants are possible, but by some reason, they are very, very rare. So by some reason, evolution made aging very, very evolutionary stable. And we believe evolution made it to prevent inhibiting aging by pathogens. So if aging is indeed an adaptation to limit epidemics, then pathogens want to make us living longer, counterintuitively, because they want us to live longer and spread pathogens as long as possible. And such experimental evidence exists. So there are ants. In ants, the queen lives like 50 times longer than workers. And some parasites can indeed make workers, infected workers, to live as long as queens. So they actually switch this developmental program, already in adult formed worker, they can switch it to the slow aging, which matches the queen longevity. And this is very clear for what pathogen needs it, right? It wants this long-lived infected ant to be eaten by a woodpecker and prevent it from dying. And idea is that evolution, the ant doesn't want that. It doesn't want to help pathogen to spread. And the idea of our model is that evolution builds some safety harnesses that prevent manipulation of aging with pathogens. And these safety harnesses may protect against mutations and, unfortunately, against therapies. So, with this, we resolved both the classical questions. We believe there is no scientific reason to believe in entropy as a source of aging anymore. And of course, this is a big ambition because in this case, aging should be reclassified from this group of ideas about physiology, metabolism, nutrition into the ideas about immunity and infection. So, and the idea meets a lot of rejection. And now we work on two connected projects. One is aimed to change the mindset, and the second one is how to use our theory to provide some meaningful results that are probably good for therapies. So idea in the changing mindset projects is that we can rationalize phenotypes that classic models cannot rationalize. So for example, why everything what flies lives so long? Why naked mole rats have negligible senescence? Why queens and eusocial insects living so long, regardless having the same genome as workers? Also, why this is not related to aging, it's related to death, but why do some animals die shortly after reproduction? And we believe that these answers to these questions are all about infections and population structures. So that we believe that we can find answers to this in epidemiological models. So idea is that in flying animals, we have low viscosity, that relatives are interacting less, and this weakens the selection, that queens are a small proportion of the population, and they are not important for epidemiological situation. For mole rats, it's a little bit more more complex. We believe that mole rats, with their eusocial structures, they develop hypersensitivity, like as you may remember, this ABI system in bacteria, that if the first animal dies in response to infection, then it is much more superior way to control pathogens than aging, and then aging can become redundant. And indeed, there are some case reports that indirectly support the idea that mole rats can be indeed hypersensitive to infections. You can also double check our recent preprint. We investigated the phenomenon that while some animals can rejuvenate, many of them avoid to rejuvenate unless under stress. So the question is, if I could rejuvenate, why should I choose death? And the classic models cannot answer these questions, and we show it mathematically, just as a theorem, and we show that this phenotype, if it is true, then it should it proves the inconsistency of the classic models, and we can resolve it this conundrum with our pathogen control model. So now a brief overview of what we do on the wet lab side. So we believe that cellular senescence. I'm not sure I need to introduce your audience to this phenomenon. So cellular senescence may be also an antiviral thing. So because right now, while many people study cellular senescence, this is exciting phenomenon, and many exciting data is there, but still not clear what for these cells evolved. People say they are involved in tumor suppression, but tumors are getting better if senescent cells accumulate. Wound healing doesn't go better with age, and senescent cells were shown to participate in embryonic development, but we find them outside of embryogenesis. And the biological function of this evolutionary conserved phenomenon isn't explained. And some people call them zombie cells, because these are cells that should be dead, but they are not dead by some reason, and staying here doing strange things. There was a small paper published almost 10 years ago that claimed that senescence is antiviral. Unfortunately, it did not got traction, but we totally believe it because it is consistent with our model. And we tried some experiments, and it seems to be so. So these are control cells, these are senescent cells. We infect them with GFP-labeled virus. And you see in control cells, there is a lot of virus replication and the little virus replication here. And this was true for many other viruses. So we tried like 5 different viruses from 4 families, and senescent cells were not good for replication also. So importantly, this effect was not because senescent cells were weak or sick. It was because senescent cells activated some innate immune pathway. We believe that type 1 interferon signaling is involved. So if we inhibit this type 1 interferon signaling, then we can help virus to reproduce in senescent cells. So typically, the virus is judged by the number of GFP-positive cells with GFP-positive virus. And typically, virus in the senescent cells doesn't reproduce at all. However, if we knock out interferon 1 receptor, or if we knock out protein STAT1, which is just downstream from interferon 1 receptor, then we can help the virus. And there are a little bit more senescent cells getting infected. And the same was true if we inhibit this STAT1 with the chemical drug. And importantly, senescent cells were producing cytokines, so that could protect healthy cells that's sitting nearby. Or like in this experiment, we just took media from senescent cells, put them on healthy cells, and viruses really hate them. So meaning that senescent cells can be indeed antiviral. Now we also see that some virus infections, and this was also shown by others, that some virus infections can induce cellular senescence. We added a little bit of granularity to these results. We showed that cellular senescence is induced in bystander cells, not in the infected cells, but in cells next to them. So this P21 means that it is senescent cells getting induced, and yellow color means that these cells are virus infected, and you see that they are in counterfeit. So, and our big model is that when the cell is infected with a virus, it starts producing cytokines to warn other cells and protect them. And but this infected cell can die, or virus can manipulate it, switch this cytokine production off. And to mitigate this, some of the healthy cells exposed to these cytokines, they go senescent. And these senescent cells are resistant to virus infection, and they produce cytokines stably, creating local memory about infection. And we pitch it as immune militia hypothesis. Like senescent cells are immune militiamen that can be called to duty from any other job. So this is where we are right now, this immune militia. This is important topic, and we have nice data that can connect us to clinical purposes, but how to cure aging? So with senescent cells, we don't think we will cure aging. How can we use our hypothesis to really help us to reach biological immortality? So the short answer is we don't know. So, but we believe that aging is part of the immune system. And this provides us with the tools, fundamental theoretical tools that we can apply. As you may know, this August Nature Aging issue was all about immune aging. But most of the people consider changes happening in immune system, and these changes are dramatic. So as deregulation, that something falls off. However, our model predicts that this is not like this. Our model predicts that just priorities of the immune system are changing, that immune system doesn't disappear, doesn't decay, it just becomes different. It starts to protect my kin from my pathogens. It can sacrifice myself to save my children. And this is a functionality to limit pathogen transmission. And this is a difference between loss of function and gain of function phenotype. Most people believe aging is loss of function. We believe it's gain of function. And here I want to cheerlead two papers. These are not very new, like maybe from 5 years ago, that indeed specific interventions that cause aging in immune cells, they induce systemic aging. So we can make mitochondria unfunctional only in T cells, or DNA damage in immune cells, and we see systemic effects, like the mouse is aging as a whole, so they also in solid organs everywhere. And so these two authors, Matthew Zavdade, we collaborate with him, and Marie Mittelbrunn, they recently, in this issue of Nature Aging, they published a review that is absolutely coherent to our theoretical model. So this, that immune system evolves from healer to saboteur. And I think this is a must read if you want to find this connector to the actual aging mechanism. So as a take-home message, if the pathogen control hypothesis is correct, aging is a programmed adaptation. We need to inhibit this program, not repair damage. Cellular senescence might be an antiviral mechanism. We believe so. And aging mechanisms must have deep connections with immunity. Immune system is predicted to contain mechanisms that evolve to damage the organism, and I believe this is the easiest and the strongest intervention we can imagine is to rejuvenate the immune system. With this, I want to thank people who work for me right now. This is my lab at CTU. Here are people who worked for me at UCSF. I developed this theory while being a postdoc, so I was funded by Raul's grant. Thank you. Thank to him for funding. These two genius students worked for me, and I'm happy to take questions.
Thank you, Peter. Very interesting. Let me ask you first, why are we always so dichotomous in the healthy longevity field? So why is it the one or the other? Why is it so black and white? Why can it not be both? Why can it be the accumulation of damage, for example, in immune cells? We see that in CD8, CD4 cells, etc. Or why would it be a programmed death through evolution or now via viruses?
Yeah, this is a good question. So I don't think that these things are mutually exclusive. This is the way how we perceive the process. And, if we speak about damage, we can find damage in every disease. Tell me which disease cannot be rationalized as a damage. In COVID patient, a lot of damages. In diabetes patient, a lot of damage. If I break a leg, then it is also damage. Just in all other diseases, we have clear reasons, and that's why we are not trying to target damage to cure this. So in aging, we just haven't seen the main mechanism, and that's why this side thing got inflated and took the place of the paradigm. So.
Are you now saying that aging is not the cause of age-related diseases?
Of course, aging is the cause of age-related diseases.
Okay.
But damage is a companion and a secondary effect of aging. But making it a cause, to me, is a nonsense. Like the same would be if we declare that damage is a cause of COVID, right? We see damage. If we mitigate damage a little bit, we can probably help a patient a little bit, like in case of aging. If we add more damage, we can damage the patient, yes. But nobody would try to cure COVID with anti-damage therapies.
Yeah. So, but if I use something, if I can show you now material I have here on my desk, but if I'm using that, then there's wear and tear, and I think tear and wear, or now it's being damaged, and I cannot actually maintain the repair mechanisms, and then whatever I have is going to decrease in function, and it's. I just show you my paper here, like. It's like wear and tear because I had it in my bag. So isn't that the same for the body? Like everything is aging, and I'm not sure if there is a genetic program in this paper. I just showed you.
Yeah. I think this is a fundamental difference between open and closed systems. So the entropy should increase by only in closed system. So, and in principle, we have very nice repair mechanisms that repair us day by day. And the question is if they are underdoing and the damage accumulates, or they are switched off at some point and allow damage to accumulate. And in first case, we need to make them more efficient and help us to repair damage better. In second case, we need to figure out why are they switched off. Mm-hmm. And I believe in the second. And I think all the data that we have, they indicate the second scenario, that repair mechanisms are going down with age.
Yeah, don't take me wrong. I love hypothesis, etc. It's I think the dichotomy in it. So would it then also hold for plants?
You mean like evolutionary model may hold for plants with reservations? So we received this question quite sometimes, but we didn't take enough to think about it carefully because the infections in plants this is not my cup of tea.
Yeah.
So
Okay. So I was so lucky last week to be in Vera Gonova's lab to actually hold for the first time a naked mole rat in my hands. That was an experience because I thought they were so ugly, but they're so beautiful. And but you just said, okay, you don't really see that they're aging. And of course, as a geriatrician, I saw a little bit the differences between them. So what can we learn from the naked mole rats, and how can we really disentangle if they either would be in your camp or in the damage accumulation camp, but just having very good mechanisms to antagonize ROS?
Yeah. So of course I think carefully negligible senescence. So like from the point of ecology, they don't age because they have mortality like 40% per year, and with the lifespan of 40 years, of course they have no chance to die of aging in the wild, at least workers. Queens can probably live longer, but phenotypically they may age that, but it doesn't convert into mortality. So the key experiment that can disentangle test the hypothesis is to infect mole rats with some viruses. Idea is that they should die immediately because of some immune effects or lack of immune response. And this actually has been done in one of these reports. Just it should be done at more regular, regulated, and downstream characterization should be done. This is one of the things that we want to do for sure because it's fancy. I also like exotic animals.
Yeah, so you are already in contact with the university in Rochester and actually try to do it, because if you're right, then make them immortal because of the virus. And I know that they have the colonies.
Yeah, and funny. Yes, I spoke to Vera on during RDD, and she told actually what also got me excited, that one of her colonies collapsed. So she was probably not very much excited about that, but one of her colonies got collapsed within 2 days. Like 200 animals died, and this also fits very well into the model of hypersensitivity.
Yeah.
So we discussed if we can collaborate, but it is still ongoing.
Okay. So imagine there is this genetic programming. So we did lots of jivas in the past, and we could only find genes which are more associated to diseases and not so much the genes which are associated with longevity. So it's the absence of having certain genes, like ApoB, for example. So how does that fit in your hypothesis? And I know you brought the viruses and the pathogens in, but how would that then fit?
No, so I think that, this is a good question. I believe aging should be very stable evolutionary and genetically, so a little variation. But don't you find like insulin receptors and stuff? Because I know that taller people are living shorter people are living longer.
Yeah. But if you're looking at the effect size, it's very, very small. So if you're looking at IGF signaling pathway, so you have multiple with very small effect sizes, but nobody has found yet that gene you should really have to be long-lived. And especially also if you're looking at innate or adaptive immune systems, they're there, but it's not that magic I think we found. So should we then look into the gene deserts?
No, I think I think we should look into the evolution of the immune system with age and find out if we can rejuvenate the immune system and different aspects of this. So maybe hypothesis-based approach in this case may work better than just systems biology. So as I told you, it might be that evolution developed some safety harnesses that prevent this super long-lived mutants from emergence. And then it means that if we mutagenize an aging gene, then it would lead to some other maladaptations like cancer that would kill a patient earlier. And so there can be evolved, antagonistic pleiotropy zealots would say, oh, yes, this is antagonistic pleiotropy. But in their concept, these links between beneficial and detrimental traits emerged just by chance. We believe that these links were wired in by evolution, and for evolutionary purpose. So that's why it might be why the effect sizes are not that great.
Okay. So what would be the greatest experiment you come up with to prove your hypothesis? Next, we already heard about the naked mole rats. But what would you do, which you might not be able to do, but let's think just out of the box?
Yeah, so we have quite some these same strange experiments. Like we think that we have ideas about these carnivorous marsupials that develop this similarity. It's related to adaptive death in general, not to aging, but there are also funny experiments. We believe that should be transmissible cancers in other carnivorous marsupials other than Tasmanian devils. And this should explain the evolution of similarity. I'm not sure if it makes sense to you and the audience, but there are like crazy evolutionary hypotheses. Also, this rejuvenation issue. If we can find real rejuvenation in nature, we believe that it should prove that classic models are incorrect. These types of things that we are thinking about. So you may not know there are immortal termites that are also eusocial, like naked mole rats, so I have had a discussion with a person who also corroborates the idea that they might be hypersensitive, like naked mole rats. So these type things, but I think we need to go more into the immune system and brainstorm, try to rationalize data in this model, and then maybe a great experiment will come out of this.
Yeah. I have one question about the immune system, but first the question, can we ever show that something is immortal?
Can we ever show that something is biologically immortal? Yes. Immortal, no. So biologically immortal is lack of mortality with age, right? So, lack of increase in mortality with age.
Yes, but yes. How do we actually showcase that? Because we have such a short lifespan. But independently, if we cannot see it, that's somebody, a tissue, a cell is not aging. I agree with you. Then it's more likely that at least the speed of aging is slower. So shouldn't we just get rid of pathogens to get rid of aging?
Wait, no. So in couple million years or like tens of millions of years, we probably will get longer lifespan if we get rid of pathogens. However, I have a good piece of news that we probably already did that, because our super fast evolution of longevity that, for like last 7 million years, we doubled it compared to chimp, right? We spread with chimp 7 million years ago. So I think just because of brains and ability to avoid infections and infected individuals, we just lowered the selective pressure. And I think that now we are on the way to increase our longevity even more. With traditional ways, it will take millions of years.
Yeah.
So again, aging is not caused mechanistically by pathogens. It is mechanistically caused by our immune system that wants to get rid of potentially infected individuals.
Yes. So sterile environment and all good, and maybe then we have allergies.
Well, this is already a question
Okay.
for a doctor, so I'm not sure I can give you medical advice.
I love your hypothesis. I think I'm not sure if I'm in your camp now, but I know that the audience has lots of questions. When Ayla is coming to the screen, what would you advise youngsters to read next to your book, and that one review you showed, to really be equipped with regards to all hypotheses we have?
Yeah, I have a couple of papers. Yes, you can find them on my website.
Okay.
But also you can see my videos on the YouTube. I have a small YouTube channel and put my conferences and interviews there.
Perfect. Okay, over to Dr. Ayla Hotzkits-Kuric. Over to you, Ayla.
Thank you very much for the great presentation and for the introduction. We have indeed lots of questions from audience, so I will start with the first one. Shouldn't we separate the question of the cause of aging, which could well be evolution, and the question of how to cure aging? We humans have the ability to transcend evolution, so a cure for aging might involve a number of damage repair interventions and/or metabolism changes beyond what was accessible to or adopted by evolution, right? As a question.
David Wood? No? I saw David in the audience. Maybe this I think could be also asked by him. So the yes and no. So traditionally, breakthroughs in healthcare are starting with understanding. There are a few exceptions. Like antidepressants. I think we still don't understand how our brain works, but antidepressants are very effective. I'm not sure that this blind approach would work in case of aging, because aging is much more complex and the experiments take much more time than with antidepressants. So I think the understanding will shift the field from really alchemistry, where we are right now, to a mature field that will solve aging and an engineering task, like it happened with the infectious diseases after Pasteur's work. I don't claim that my model is that one, but I feel that understanding is, I believe in my model, but I believe that understanding is necessary to cure aging.
Thank you for the answer. I will go to the next one, and it's also an interesting one for me. So given that the senescent cells can protect healthy neighbors from viruses through their secretions, does removing them with senolytic therapies risk increasing susceptibility to infection, especially in older adults?
So I think that generally, so this is a good question. I don't have exact answer. I believe the benefit of senescent cells in infection is while the infection is going on there. After the infection is resolved, they should be removed. So we probably need to avoid senolytic therapies during the time of infection. With these senescent cells that are accumulated in adults, these can be different senescent cells because they are not induced by the virus. They're induced by other means, and they can have fundamentally different properties. So we are studying that, but so far we studied only the virus-induced senescent cells and chemical-induced senescent cells. We didn't make much with the age-induced senescent cells. So that's why it can be both. I cannot answer this question.
So how do you think this can actually influence the approaches to therapeutics and current use of xenotherapeutics?
Yeah, so basically now we are focused mostly on symptoms of the viruses that may be associated with post-infection accumulation of senescent cells. How can it be used for senolytics? In the future, we want to instruct our research with virology if senescent cells are really antiviral, then viruses should, for example, make inhibitors that can open new avenues in development of senolytics and xenomorphics. So they will help us to understand mechanisms with their evolutionary wisdom.
Thank you for the answer. So here's an interesting question. Aging happens after the fertility window of women. So how does the evolution exerts its effect on aging? Do you suggest that the aging program is inherited from males?
Wait. So the question is why males are living shorter than women?
Yeah.
So this is a good question. So this is also one of the questions that our model can potentially answer. So the idea is that in mammalian population, most of the pathogens of concern are sexually transmitted. So I remind you that pathogens that aging probably evolved to control are like syphilis, gonorrhea, HIV, these things that are not giving opportunity to reproduce, but don't kill. And these are mostly sexually transmissible. And in this situation, the gender that has more sexual contacts with multiple partners potentially is a super spreader. So that if in polyhamous species, males are living shorter. Because males are, so according to our model, males are super spreaders. And the idea is that, and in birds, which are much more monohamous, both genders are living more or less the same. And the idea that in humans, actually, the difference between longevity in males and females is smaller than in many other mammals. So, but this effect is still probably because human males are sometimes, practice infidelity and are potentially super spreaders. Evolution wants to remove them.
Okay, thank you for the answer. So how might your work influence our understanding on chronic viral infections like herpes or COVID-19?
No, well, COVID-19 isn't a chronic infection, so unless
chronic
in very few cases.
I think, I think the question was based on the chronic COVID-19.
No, well, I think chronic COVID is a little problem. So the herpes, genital herpes can be part of the model because it can sterilize. So the herpes, which is on the on the lips, so it also can be of interest because also the model predicts that chronic inflammation or chronic infection should accelerate aging, age-related disease, and this is of course not very honest prediction because everybody knows this is true. And as you may know, people with herpes, if untreated, they have earlier onset of Alzheimer. And this is very in line with the model, specifically its earlier stages. It inspired me a lot so that this fits the prediction that chronic inflammation, okay, the body feels that we are probably chronically infected, we need to kill ourselves faster.
I understand. And from a diagnostic standpoint, what would you consider the most promising biomarkers of pathogen-driven aging phenotype? Immune signatures or transcriptomics, inflammatory cytokine panels? How we can translate your findings into clinical practice?
Yeah, thank you very much for the question. I think this was not a misunderstanding in this question because I don't believe pathogens anyhow drive aging. So I believe aging is part of the immune system, that this is from our host side, the mechanism that removes potentially infected individuals. It is sort of a, if it is can be said here, not very gently, prophylactics. That the immune system thinks that at certain age, we have high probability to be infected with bad things. And then it kills us. So in terms of biomarkers, again, I'm not sure I can give a top-notch answer to this question. It's probably Dr. Maier would be 10,000 times better than me, but I believe immune system and cytokines, they reflect process of aging quite well. But here I'm not original.
Okay, let me see. Anything else? So suppose that aging is indeed a programmable and evolved for a reason. How do we ethically and biologically approach turning it off without disrupting the immune balance that has protected us for millennia?
Immune balance. Well, so here is the idea that there is a young immune system that protects us, and this old age immune system that protects our kin. If we find a way how to prolong or rejuvenate immune system, we can turn the old people back to their young immune status, and it will be they will still be better protected from pathogens than they are now in the old ages. So I don't think that there will be any problem, ethical or other, if we can do that.
Okay, thank you very much for your answers. I think there are no more questions.
Thank you so much. I really enjoyed this discussion, and I fully agree with you, Professor Lidsky. The immune system is so important, and avoid the blisters on your lips. Yes, absolutely. Thank you so much, Dr. Eiler-Hodgkiss. Thank you for staying and being here. We have a couple of announcements to make. Of course, we have organized a couple of conferences for you in Singapore. November 17th to 18th, so that's next week. The Healthy Longevity and Medicine Conference is happening at NUS UTown, co-organized by the NUS National University of Singapore in the Asahi Labs, Japan, and supported by the Embassy of Japan in Singapore. This conference is really focusing on how to bring healthy longevity into public services, and tips and tricks. Please use the QR code to get more information. Then the second one is the Healthy Longevity Intensive Course in the conference. It's happening February 26th next year, from the 23rd to the 27th of the second month of that year. It's organized by the NUS Academy for Healthy Longevity, 3 days of intensive course and 2 days of conference, followed by an open day for the public. And of course, abstract submissions are still possible. And if you have a startup, please use this podium to pitch. Our next Healthy Longevity webinar is on the 20th of November, and Associate Professor Kaiko Konoi will be there talking about plasma membrane damage and aging. As always, we are leaving you with a little teaser for our next episode. Take care.
I'm trying to identify the chemical genetic intervention that delay the aging and increase the lifespan. So I'm using the very powerful model system yeast for cellular aging.
So what makes yeast such a powerful tool in understanding aging?